We're going to get started. It's a great pleasure for me to introduce our speaker today, Denise Ramires, a current student in the population biology graduate group, also a member of our lab. Denise and I have a lot in common. We're both Colombian. We both went to the same undergraduate institution. We both have the same last name. We're not related. We were all related. Go back in time.
Our most recent common ancestor.
So, and the one thing that we also share in common that is the reason for Denise being here is that we have a big passion for bees and brains, which is what Denise has been working in the lab. As you know, our favorite animal to study in our lab is the orchid bees, and in particular, this one species that lives in Florida that has enabled us to study a lot of the variation in behavior, mating behavior, social behavior. And Denise joined the lab kind of at the right time. You know, we've been doing all these behavioral and genetics studies, and we've been dreaming about looking under the hood and what happens in the brain. And Denise, when she was completing her undergraduate thesis, she did a really nice study in the morphological diversity of particular bees, which is the clade that includes bees. And so Denise came with the right questions and a lot of techniques that she already knew in terms of morphology and histology. And it's been really phenomenal to have her in the lab developing her project on understanding how the variation in behavior that we observe in males and females correlates to variation in brain plasticity. So I'll also mention that Jason baked some delicious cookies with brains. Color brain. So as you learn about the different parts of the brain through the talk, you can just start, eating them. So, Denise, take that away.
Santiago, thank you very much for your introduction, and thank you everyone for being here, either in person or in Zoom. I'm very excited to be sharing with you some of the work I've been able to complete so far, and some of the work that will be following in the years to come, and everything encompassed under my title, Untangling the Relationship Between Behavioral Diversity and Brain Evolution in the Orchid Bee Euglossa dilemma. So I want us to start by thinking about the vast diversity of insects on Earth. So insects alone encompass more than 60% of the total diversity of extant animals. And this diversity has been accompanied as well with a wide behavioral and ecological diversity. So we have, for example, I'm going to use the ruler here, dung beetles from arid deserts to butterflies that have amazing migration journeys, to animals that live in aquatic environments, and to honeybees that have social organizations. And so many of these behaviors are supported by remarkable cognitive abilities that resemble those of vertebrate species, such as long-term memory formation, associative learning, and social communication. So here we have just an example of some primates having social learning, and we have an analogous example of honeybees that communicate with each other the place and direction of sites that are basically good for food resources. And some insects have even pushed these abilities farther beyond the human brain and are able to integrate information that is inaccessible to our own brain. So for example, we have a very cool example with nocturnal dung beetles that are able to use the light of the moon as a sky compass to navigate basically this arid desert. And some of these species are even able to collect more than 250 times their own weight in dung in just one day. And they basically use these resources for their survival and reproduction. And very much like invertebrates, we can trace this behavior back to the brain. So here in this panel, we have the brain of different vertebrate species, mammalian brain and reptilian brain. And here we have basically color-coded different regions of the brain by specialized functions. And we have here in the other side, basically the analogous of the vertebrate brain in invertebrates brain. Here are two examples of arthropods. Here we have the insect brain, and here down we have a decapod brain, also color-coded by specialized regions having specialized functions. And so the fact that the brain has functional specialized regions facilitates the study of behavior in the sense that we can start linking certain behaviors to specific brain regions and understanding how these brain regions might be changing, correlating to behavior. And so here is just an example. Basically, you are visualizing the visual part of the brain, color-coded in all our species with color yellow. And so basically, when we look at the brain of the insect, it looks something like this. We have visual regions color-coded in yellow. We have olfaction brain regions color-coded in fuchsia, and multisensory high-order processing regions color-coded in aquamarine. And the interesting thing about this is that we know that modifications in different brain structures can inform sites of adaptive behaviors. So here, as an example, we have honeybees. Honeybees are extremely visual organisms that rely on different visual cues to communicate to each other in their social organization. And what I want you to notice here is that these two lobes, the medulla and the lobula, the names are not important in this case, are basically optic lobes that are encompassing a big proportion of the total size of the brain in honeybees. And these regions are basically integrating visual information from the environment. And contrary to the honeybees, we have organisms such as ants. So ants, when you think about an ant, they are mainly subterranean animals. They have basically lost many of their sight and most of their communications, and basically they sense the world via their antennae, basically chemical communication. And when we look again at the structure that we were comparing with honeybees, we see an immense reduction in volume in these structures. Basically because neural tissue is expensive. So why invest on something if you really don't need that for your survival or reproduction? And so basically studying the brain can help us inform the basis of behavioral traits and vice versa. Because if we know and understand a behavior, we can then study the brain and start correlating behavior with brain regions. And at the same time, if we understand the function of different brain regions and we know behavior, we can understand the basis of different behaviors. And that leads me to my study system, as Santiago was mentioning, the orchid bee Euglossa dilemma. I call the species a system of opportunities, not only because we have an extensive literature on life history, behavior, and evolution that has been possible thanks to the extensive work in the Ramires lab and collaborative labs, but also by the fact that this species has a very well documented dichotomy in its behaviors. So we have females that are primitively social, that you see here, they create little social colonies. I will explain this behavior in just a second. And then we have solitary perfume collecting males. And we have access basically to both of the sexes easily, because yet again, as Santiago was mentioning, we have a nice geographical area where we can study these populations. And we have also the flexibility to study these species, both in the wild and in captivity. So we have a very nice system where we can basically monitor our species in the wild by cameras that we can place on wooden boxes that these bees are gonna colonize in the wild, as you can see here in the video. And then we can monitor how these behaviors are basically progressing through the life of the bee. And we can also then study them in captivity by some greenhouses that we have built in the University of Florida. And basically we can do their behavioral experiments and then try to understand different modifications in brain regions correlated to the behaviors that we're trying to study. And you may think about orchid bees, neotropical bees. So generally they are distributed from Mexico, this species from Mexico all the way to Costa Rica, but they were accidentally introduced to Florida some years back, establishing a very healthy population that allows us again to have easy access to these species to continue studying it in long term. And just as I say, I'm going to give a brief overview of how the social system of this bee works. So females, some of the females are going to emerge, and some of the females are going to decide to disperse, and they're going to become a foundress female that is going to be characterized by the foraging for nectar, pollen, and resin to start building these brood cells. After building the brood cells, she's going to basically stop every foraging activity. She's going to stay basically in the nest. She's just going to go a few times to forage for nectar, basically just to keep her metabolism going. She's going to stay in the nest, basically, as the name says, guarding those brood cells, making sure that predators or parasitoid animals don't enter that nest and attack its brood cells. And then after some of the bees start emerging, some of those females are going to disperse, repeating the cycle, but some of those other females are going to decide to stay in the maternal nest. Those females that stay are going to become subordinates. And then our foundress now becomes a dominant bee, which is basically, again, just staying in the nest, kind of monitoring those brood cells. And now the subordinates are going to be the ones that are going to be foraging for nectar, for pollen, for the next generation of bees. And then we have the males that, as we mentioned before, are solitary, and they are very cool in the sense that when we think about animals, we generally think about pheromones, and like animals synthesize their own pheromones, and then they express these pheromones to attract conspecifics. In the case of orchid bees, males don't synthesize their own pheromones, but they rely on natural resources that they're going to basically collect to create a perfume blend that is species specific. And then they're going to use this perfume blend as a pheromone analog to attract females. And they also display behavior that is called site fidelity. So males have a wide range of foraging and home range, and they're basically able to have a memory trace of sites that are good for nectar, and they're basically gonna repeat those same foraging sites in order to facilitate this behavior. So like after seeing this, you may think like, these are very complex behaviors. So the fact that we know a lot about the life history of the species and combining the complex dichotomy of the species, it makes it a very appealing system to study the relationship between brain and behavior, especially because studies at the intraspecific level can provide us unique opportunities to do this for two main reasons. One is that we can study differences at the species level. So for example, comparing males and females, how in one species we have two very distinct behaviors. How is the brain basically flexible to adapt to each of these behaviors within the species? And then also at the individual level, to understand the brain adaptability to new behaviors and environments. So in just one organism, how is the brain able to adapt to new cognitive demands as a bee ages or gains experience? And this leads me to my research questions. So for my chapter 1, I'm asking, is the brain of males and females anatomically dimorphic? And I will be approaching this using micro-CT scanning and confocal microscopy. In chapter 2, I'm asking, how is brain plasticity supporting key behaviors in Untangling lemma? And I will be approaching this question using imaging and behavioral experiments. And then finally, in chapter 3, I'm asking, can the cellular composition of the brain explain behavioral differences between the sexes? And I'm going to be targeting this question using single-cell sequencing to basically characterize the different populations of cells in the brain between males and females. And so let's start with chapter 1. Is the brain of males and females anatomically dimorphic? This is the chapter that I'm gonna most dwell in. It's the chapter that I have more data right now. So please bear with me while I tell you the story. And then second and third chapter are gonna be quite short, but hopefully in the years to come, I'll be able to give more details about it. So for this first chapter, I'm basically reconstructing three-dimensional sex-specific brain atlases. And what I'm doing with this is basically creating volumetric comparisons. So as I mentioned before in the introduction, we know that modifications in brain structures can be informative of sites of adaptive behaviors. And we know that behavioral and sensory adaptations are often reflected in differential expansion of brain components. So again, I'm going to study different brain regions. Here we have color-coded again our different types of regions, optic regions, olfaction regions, multisensory regions. And basically those are just the list of brain regions that I'm interested in comparing. And then I'm going to be complementing this volumetric analysis with data on neural connectivity, because it's not only about the size, but how those wirings and how those connections are being integrated in the brain. And I'm going to study these using confocal microscopy to study and quantify specific microglomeruli. So microglomeruli is a weird word, but it's basically just microcircuits in a specific part of the brain of a bee, called the mushroom body, which is the center for learning and memory. And a very cool thing about the mushroom body of bees is that it's subdivided in subcompartments that allow us to understand how different inputs are being targeted in the brain of the bee. So here we just have basically an histological slide of the mushroom body. And what we can see here is that we have a discrete region known as the lip. And the lip is receiving all afferents from the antenna. So it's basically a region that is receiving olfaction information. And we then have the collar, which is basically a subregion receiving only afferents from the optic lobes. So we have two distinct parts in a very complex structure that we can then analyze and quantify how different neural connections are differentiating between males and females. And some of the results I've been able to gather so far are in my volumetric comparisons. As I mentioned before, I'm using micro CT scanning. So basically, it's a technique that uses X-ray sources to basically create an image based on different contrasts. And I need to give a special thanks here to Douglas Rowland at the Genome Center, who has been basically my micro CT guru in my learning technique. And so what I'm doing is that I'm taking bees that are less than 36 hours of emergence to control for any aspect of age and experience in this group. And I'm using an iodine solution to enhance the soft tissue of the brain in order to have good contrast when I'm scanning. And I'm using minimum 10 individuals per sex in order to do my analysis. So we then basically just put our little brains in the micro CT scanning, and we can have then three-dimensional reconstructions of our regions of interest. So our raw data is gonna look something like this, but then we can basically process this data, segment our regions of interest. So here right now, please look at your cookie. These are the color codes in your cookie. Thanks, Jason, and thanks, Colin, for this amazing gift. And then after we are able to segment our regions of interest, we don't only have three-dimensional reconstructions that allow us to see in exquisite details anatomy that we have never seen before in 3 dimensions, and also understanding how it's basically inside the head capsule. It's not an isolated structure, but we can see it in basically how it's organized in the head of the bee, and also understand different aspects of the connection of the brain and how it's working and how it can differentiate between both sexes. And so basically, after having this and having our volumes, what I'm doing is I'm characterizing patterns of allometry and relative investment. So I'm not going to go into the details of the equation. I just want you to know that we're interested in the intercept and the slope of this equation. So the intercept is going to be our proxy of relative investment, and our slope is basically going to be our allometric scaling. And we're always going to be scaling our region of interest to a part of the brain that we're going to call the remaining central brain, which is this structure in gray here, which is going to be constant in every single comparison to make comparisons viable and comparable between each. So why study allometry? Allometry is important because it allows us to describe and interpret the evolution of morphological variation. So we can have basically 3 different cases. The first case is a change in slope, so a change in allometric scaling. If we have a change in allometric scaling, we cannot compare investment, because if 2 things are not scaling the same way, it's very hard to compare how different things are investing on one same tissue. Then we can have a change in intercept, which is basically our most interesting case, because there are 2 sexes that are basically scaling the same way, but one is investing more than the other. And this is where we can start thinking about adaptive behaviors or unique behaviors that are basically correlated to one sex or another. And then we have basically just shifts a same allometric baseline, which is basically just scaling in the same way, but if you're bigger, then everything is bigger. And so when we do our analysis, the first thing that we find is that males invest more in all primary optic regions compared to females. So here in our graphs, we have in our x-axis our remaining central brain, and in our y-axis our regions of interest. And what is important for you to see here is that in all of these cases, the yellow line that is representing males is always above the purple line that is representing the females. So again, as you might remember, I was saying that the intercept in this case is the proxy of relative investment. And so we can say that males are investing more than females. And the hypothesis is the fact that males forage later in the day than females, and they also have wider home ranges. So females are very local and very loyal to their foraging sites, while males tend to have wider ranges that can go up to 20 kilometers. And they also are found later in the day foraging for nectar, probably because during the day, they're also searching for these perfume compounds that they're gonna use during this play. And so these made me interested in testing other senses that we're usually not accustomed to think about. So basically, I'm interested in the role of polarized light in navigation. And so what is polarized light? Polarized light is basically the light of the sun, that once it hits a specific structure, it changes the direction of its vectors, and it creates a specific pattern of light, that can be recognized by insects in two main structures that are the ocelli and the dorsal rim area. So the ocelli are simple eyes in insects that are basically this little 3 dots here. And the dorsal rim area is a specific part of the compound eye of insects that is specialized in perceiving these types of lights and these types of patterns that are quite unique and important for navigation and for sky compass orientation. And fortunately for me, these are both structures that we can also segment using microCT. And when we segment and compare again the volume of these two structures between males and females, we start seeing the same pattern as we saw with the other optic nerve pulse. So in both cases, we have the line of the males above the line of the females. So we have a higher investment in this tissue in males than in females. And this basically further suggests the importance of visual cues in key behaviors for males. The other thing that we found is that females invest more in the mushroom body than males, and mushroom bodies are basically the center for learning and memory in insects. Even some authors have described the mushroom body as the soul of the insect. And what we can see again is the same pattern of graphs that we're accustomed now to see. In our x-axis, our remaining central brain, our allometric control, and in our y-axis, our region of interest, which is in this case, the mushroom body. But the mushroom body is a very complex structure. It's divided into 3 different subregions. The input region is known as the calyx, which is here color-coded with aquamarine. And the calyx is going to be basically composed of dendrites. So all the afferents and all the information coming from different optic lobes of the brain are going to target this area of the brain. And then we have a pedunculus and the lobes. So the pedunculus basically are the axons of those neurons that are going to then bifurcate in an alpha and a beta lobe, which is the output region of this brain. Yes, the output region of this brain region. And it's also the part where memory consolidation occurs. So we can go ahead and basically segment each of these parts by its own and ask if the differences that we're seeing between males and females are being driven by a specific subregion of the mushroom body. And the answer is yes. It's driven by the input region of the mushroom body, the thing that is driving the differences between males and females. So again, when we see the regression line for the calyx, we see that the line of females is above the line of males, even though they share a same allometric scaling. While the pedunculus, there is really no difference between males and females. So what this is telling us is probably that females are having higher cognitive demands than males, and so they need to have a wider input region that allows them to target all that information that is being perceived by the environment. And as you might remember from my introduction, this very specific region is subdivided into sensory modalities subcompartments. And we can go ahead and further segment these regions to understand if there is a specific sensory modality that is differentiated between males and females. So we segment the lip, the color, and the basal ring, the lip from olfaction, the color for vision, and the basal ring, which is an integration point of both signals. And when we run our analysis, we find that only the lip is actually different between females and males. And olfaction, it seems then, that is playing a key role in the volumetric differences seen between the sexes. And my hypothesis is that this is linked to social behavior. So we know that in social insects, communication is mainly driven by olfaction, by chemical signals. So we can see again, 2 examples of orchid bees, and we can see that they are using a lot also their antennae to basically communicate to one another, but also to take care of their root cells. So the mushroom body is basically a brain region that is very dense in connections, and is a brain region that it has unique neurons called Kenyan cells. And mathematical modeling has suggested that higher number of Kenyan cells indicate enhanced cognition. So I asked, do the larger mushroom bodies in females also correlate with a larger number of Kenyan cells? And the answer again is yes. When we plot again our absolute volume of Kenyan cells between males and females, we see a clear difference between the volume of this cluster of cells. And now the important thing or interesting thing to think about is that maybe volume is not a real proxy of cell number, because what if the compression of the cells is different between the sexes? So, oh no, give me 1 second. Just sorry about that. So again, the compression of the neurons may be different between both sexes, right? So what we're trying to do right now, this is an ongoing project. I don't have definite results for this so far, but it's exploring novel imaging avenues to target novel questions. So this is a project, again, in collaboration with Douglas Rowland at the Genome Center and Elizabeth Clark, a postdoc in the University of Berkeley, where we're using synchrotron technology to scan at higher resolution. And we're able to then segment individual cell neurons to basically quantify in an honest way the total number of cells between males and females to have a more robust and confident answer to this question. And so to start wrapping this chapter, the one last thing that we need to have in mind is that when we go ahead and study the brain body allometry, in both sexes, this is conserved. So both sexes basically share the same allometry. And when we plot this allometry and compare it to different allometries that have been reported for different bee species and Hymenoptera species, we see that actually Euglossa dilemma fits quite well in the reported scaling coefficients that have been reported for other bees. And so the fact that both males and females share a common allometric scaling is telling us that allocation rather than investment best explains the volumetric differences seen between both sexes. Because they are investing in the same, like in broader scales, they are investing the same in neural tissue, but they're basically allocating more to one region or another, depending probably on their behaviors. Okay, so now let's jump to chapter 2. How is brain plasticity supporting key behaviors in Euglossa dilemma? So first of all, what is brain plasticity or neuroplasticity is basically the ability of the brain to change throughout the individual's life, enabled to allow it to cope to different cognitive demands or novel environments. And we can have two types of neuroplasticity. Age-dependent neuroplasticity that, as the word says it, is going to depend on age. So a very neat and cool example is found in honeybees. So honeybees, when they're born, they're going to stay in their colonies for a couple of days. And they're basically going to work as nurses, just taking care of the brood cells, right? But then after a very specific time, these nurses are going to become foreigners. And just before that transition occurs, the mushroom body, this area of the brain, has an expansion. And this expansion is basically correlated by the fact that these bees are not going to be any longer just inside the colony, but they need to face the novel environment outside the colony. They need to start foraging, they're going to need to start integrating new signals that are novel for them. So this is age-dependent neuroplasticity, and it basically functions to prime the brain to novel behaviors that are basically anticipated. And then we have the other type of brain plasticity known as experience-dependent plasticity. So in this case, we could have, for example, one bee that as it forages and forages and forages, the same area of the brain known to be involved in learning and memory also starts increasing further its size by the fact that it's being stimulated constantly. So age-dependent neuroplasticity is not going to be chronological, but it's going to depend on the experience. You can have a bee that can live a very long age, but if she's never basically faced with different cognitive demands, she may not develop this type of brain plasticity. So how to study neuroplasticity? As you can see, there are many factors that can affect basically how brains can change throughout their life. So what I'm going to try and do is dissect these complex lifestyles into smaller components. And for this, I'm going to have 4 different treatments. I'm going to have wild bees, and then I'm going to have 3 different treatments that are basically aimed to restrict just one type of influence in brain plasticity. So age, social interactions, and cell collection. And I'm going to be also using the same modality as in chapter 1, micro-CT and confocal microscopy, in order to be able to compare it to our basic baseline, which is chapter 1, that as you might remember, there are naive bees, and we're controlling for any age or experience in chapter 1. So experimental design and progress. So for wild bees, I'm gonna basically be targeting the social phases of the females and then getting wild-caught males that are able to forage, are able to reproduce, and are able to collect scents. And during the summer, I was able to target the foundresses, the guards, and all wild bees. Then I'm asking the age-dependent treatment. So these are bees that are gonna be in isolation and are gonna be allowed to age for 10 days. Basically, I will be providing honey water for them. They don't need to forage. They don't need to have any like cognitive demands in order to find their food. And I'm aiming for a minimum of, again, 10 individuals per sex. And so this summer, I was able to collect 13 females in isolation and 3 males. I hope that the next years to come, I'm able to finish my male treatments. And then for the social treatment, we're asking if social interactions are enhanced by brain plasticity. And the way I'm targeting this question is having bees basically coupled with another bee and just allow them to interact with one another for 10 days. So we're gonna again be controlling for the age because we already have an age treatment for 10 days. And any posterior change that we see in the brain, we can then attribute it to social interaction alone. Because again, I will be the one providing honey water for these bees and they don't need to go ahead and basically have the demands of their environment. And so far I've been able to do 6 treatments of the bees. These are paired bees. So basically I will randomly choose just 1 bee of each treatment. So my data are able to be independent. And hopefully again, during the years to come, I'll be able to finish my treatments for both females and males. And then last, for scent collection, I will be basically allowing males and females to be exposed to scent for 10 days again, in order to control the age treatment that we're gonna be having. And then any posterior change that we see in the brain or in a specific area of the brain, we can then attribute that change to scent collection as basically the input force that is driving brain changes. So there are a lot of treatments, so you can ask, where do we begin? And I decided to begin by the extremes. I'm basically comparing newly emerged males that are the ones in chapter 1 with wild-caught males. And here I need to give a special thanks to Marisa that is joining me here today, and to Denise, a former student in the Ramires lab, because with help of them, I was able to collect my samples. And in order to try to have a little bit of control on the wild-caught males, because we have a range of ages and experience in the wild. What I did was collect males that had a full capillary of perfume as a proxy of age and experience, because we know that for a male to have a full capillary of perfume, it takes at least some days or even some weeks, in order to achieve this. And then it's also a proxy of experience because it's a male that has successfully been able to collect this complex perfume blend for reproduction. And when we do the analysis, again, using micro-CT, I'm talking here about volumetric differences, we find that wild males have an enlarged mushroom body compared to newly emerged males. And so this difference is mainly driven by what we know as post-eclosion dendritic growth. So it's basically our dendrites in a region that are expanding and creating new connections instead of just new cells being created. And we know this because when we go ahead and again measure the absolute volume of Kenyan cells, we find that there is really no significant difference between both of our treatments. So again, this further suggests that males, some behaviors of males are basically enhancing and demanding some memory and learning consolidation throughout the bee's life. The other thing that we find is that wild-caught males have an enlargement in a visual region called the anterior optical tubercle. And this is important because this region is basically receiving afferents from those 3 optic regions that I mentioned in the first chapter that we find to be enlarged in males compared to females. And this little brain region that, although it's very little, plays a very important role in the circuits of polarized light and color learning. So this further suggests, again, the importance of visual cues in the survival and reproduction of males. So the coming work for the years to come is basically lots of segmenting and frustration, but I hope that as I go and study more my treatments, new and novel results are going to start to come out. And finally, my third chapter, can the cellular composition of the brain explain behavioral differences between the sexes? So far, we've been talking a lot about morphological differences and anatomical differences and brain circuits, but I want to untangle the relationship between brain and behavior in a multi-level approach. So the idea now is to build basically a cellular sex-specific brain atlas that will allow me to compare different clusters of different cell types between both sexes. And the way of doing this is by single cell sequencing. So some of you may be familiarized with this technique. Some of you might be saying, what is single cell sequencing? So basically, it's an emerging technique that allows us not to just have transcripts of general expression of genes, but also to cluster these transcripts into different populations of cells. And so it's important because we can then compare expressions within cell types and between cell types, and it can even allows us to basically distinguish novel profiles of populations of cells that can also then be linked to quite unique behaviors between the sexes. I don't know if this is something that we're going to find or not, but it's basically my motivation behind using this technique. And then after doing it, we're hopefully going to obtain something like this, where we can have a characterization of the different populations of brain cell types between males and females. And once we have that characterization of cell types, we can then even have a further level of detail in the comparisons between brain and behavior in the orchid bee. And this is a project that I hope is in collaboration with Steve Montgomery at the University of Bristol. They are currently working on some projects using this technique and basically creating a faithful pipeline to analyze this data that is a little bit tricky. So I hope that the years to come will allow me to make this come true. And so just as a brief recap, in chapter 1, I asked, is the brain of males and females anatomically dimorphic? The answer is basically yes. And there are still a lot of things to explore that I hope that I can give a little bit more resolution once I start incorporating the data of confocal microscopy and understand how that neural circuit may also be differing between males and females. Then in chapter 2, I asked, how is brain plasticity supporting key behaviors in Euglossa dilemma? So again, for this chapter, I will be using behavioral experiments to target unique behaviors. And then chapter 3, I'm asking, can the cellular composition of the brain explain behavioral differences between the sexes? And with that, I just want to give a special thanks, first of all, to Santiago. He has been an amazing support since day 1. Thank you for opening me the arms to the lab. Thank you as well to the Ramires lab, and a special thanks to Kathy, that, I'm gonna cry. She was a postdoc in our lab, and she left today, but I'm so grateful for her. To my QE committee, Rachel Bay, Kayla Sowski, Peter Wainwright, Phil Ward, and Steve Montgomery at the University of Bristol, to Doc at the Genome Center, to the EVA lab at the University of Bristol that I had the chance to visit earlier this year and learn a lot. To a lot of people that have been an amazing support so far, and that I'm pretty sure that are going to be an amazing support in the years to come as well. To my family, Javi, my partner that is sitting right there, and my cats, to the bees. I've decapitated a lot of them. Thank you all for being here. And now with my remaining central brain, I'll be happy to answer your questions.
I really enjoyed your talk, Denise. So for your single cell RNA sequencing, I'm not sure if you're using like 10x genomics or.
Yeah, so the idea is to use 10x, which I know that currently, what is that lab? Olga and Ben, Artyom's lab, sorry. Artyom's lab is currently using 10x, but I know that when you have more samples, like when you're doing bulk samples, 10x might not be the best choice. In my case, I don't plan to have a lot of samples. I just want to have a characterization of the different populations of cell types. So I think I'm going to go to 10x, but it also depends on basically what I go ahead and talk with Steve when we start collaborating in this project. Are you interested?
No, I was just because I wanted, they're usually like optimized in like human and like mice, and I was wondering, since you have like a non-model organism, do you expect any sort of challenges with?
Yeah, I definitely do. I know that 10x usually comes here to Davis and like they give talks and seminars. I went to one of the single cell sequencing, and they do indeed basically standardize a lot of things in like model organisms in vertebrate species. But they like the tricky thing is getting a high number of cells, which is sometimes hard when you're working with tiny organisms. I know in Drosophila, they, for example, need to pull out like more than 20 legs to just have one sample. But it seems, for what I've talked to Olga, that the size of a breed brain would be enough to have a good sample for single cell sequencing. But I still don't know how that's going to go. So, but thank you. Yeah.
Is there a difference in brain morphology between, I guess, the dominant and subordinate, or in more, I guess, I'm thinking like in the honeybee, is there a big difference in the brain morphology between the queen and the workers?
Yeah, that is a great question. That is actually one of the things I want to target. Just go back. In my experiments of wild populations. So basically, I want to target the founder state, the guard state, the dominant, and the subordinate states to exactly just test those potential differences. Because we do know, for example, that in honeybees, queens have a very distinct brains than foragers and even drones. So drones have a very enlarged, again, visual system, but in this case, the enlargement of visual system has been attributed by the fact that males actively search for females for reproduction. While in the case of orchid bees, that is not what we see. Males don't really search for females. They just start displaying, and females are the ones that are going to come to males. So that's the interesting thing, because otherwise, like in a lot of insects, when you see an enlargement in the visual areas of the males, are actually species where the males are the ones searching for females and not vice versa. So this is why like this might be an interesting case study to further study the role of optic regions in the basically performance of males. So I still, to answer your question, basically, I don't know yet, but hopefully we'll be able to find out at some point in the next couple of years. Yes.
My question might not even make sense, but I'm wondering is if you have a way to assess whether the male or female brain is the derived version, evolutionarily derived, which one has departed, which one, if either or both, has departed from the more primitive condition?
I think that's a great question, and I think for that we would need to have a more phylogenetic comparative approach in order to see basically how it has changed in closely related species and basically know what was our baseline there. The thing is that with brains and insects, they can change quite rapidly, and again, even within the species. So I don't know. Yeah, I don't know how to answer that. I don't know how we'd start answering that question. I think it's very interesting, and like in broad patterns of brain evolution in insects, we can see when certain things start changing. For example, we know that by the acquisition of parasitoidism in bees, the mushroom body created a huge expansion compared to the rest of insects. The rest of insects, when we look at the mushroom body, is not a structure that has a double cup. It's basically just a stalk, just like that. So you can start seeing certain patterns of morphological variation and behavioral traits in like big, broader scales. I don't know if at the level of the species, I would be able to answer that question. Yes, ma'am.
I first wanted to just congratulate you on such a huge amount of work done already. The chapter 1 is huge. Second, Yeah, I don't know if this will connect with you, but in my undergraduate research, I worked on a project that found that in sleep deprived flies, the mushroom bodies and especially the Kenyan cells demonstrated like more synapses after sleep deprivation, especially like out of all the brain regions, those really popped out. Does that connect at all with the sexual dimorphism you see between males and females? And I don't know, maybe like, is there one sex that is just chronically more sleep deprived than the other?
I think that is a great question, and I think I will be able to answer that once I get into the confocal microscopy, because when we're talking about those brain connections, especially in the mushroom body, we're talking about the microglomeruli that are basically, once you stain the bee with antibody, you can basically quantify those points of contact because they create these strict buttons of synapse. And the interesting thing is that they are quite variable into what changes depending on what treatment. So for example, it has been found that in honeybees, foraging creates an increase in volume, but it creates a decrease in synaptic connections. And this has been basically attributed by something called synaptic pruning. So basically you get rid of all those connections that you don't need and you enhance those that you do. And those are the ones that basically branch and create that volumetric difference. I'm guessing that in deprived flies, I don't know what's going on. Maybe they're just like trying to struggle with, I don't know if they deprive it with light or if it's stress that happens to just cause that. Because in isolated organisms, also sometimes what we actually see is a reduction in the mushroom bodies. So they basically get atrophiated by the fact that they're not being able to really accomplish their interactions. And it's something that is quite interesting because even when we think about humans and behavior in humans and how the brain basically changes in humans in isolation, we can also start seeing some distortions in the brain. So I think it's basically a very interesting parallel of how studying such simple organisms can also allow us to understand more complex behaviors and patterns, even in our own species. So yeah, I'm sorry, I know I didn't answer your question, I just dwell on it, but I really don't know what's going on there. Yes.
Yeah, that's great. I was curious, like, you kind of hit on this with, you were saying that you were looking to do like, like density of cells and not just like the volume, for instance.
Yeah.
And I was curious, I know in like vertebrate-based neurobiology, like a lot of it also has to do with things like surface area and folding. Is that like a similar story in invertebrates, or is it really a lot more about like that kind of, like you were saying, like the compression as opposed to like the intricate folding and like that ratio between the surface area and the volume?
Yeah, that's actually a great question because the mushroom bodies are actually what's being analogous and considered as the cerebellum, like an analogous structure as for the cerebellum. And it has been, I don't know if you're familiarized with the primate brain, the social brain hypothesis that basically states that the brain in primate has been expanding in the neocortex as organisms get more socially organized. And in this case, what we see is that, again, that structure called the mushroom body, for example, in Drosophila, is just basically like, this is like to say the pedunculus, and we just have here a ball that is not even like a cup-like structure that supports cells. It's just a ball. So when you measure that surface area, it's really not a lot for cells to basically be anchored to that neuropile or that brain region. Well, then in the mushroom body, what happens is that that ball gets invaginated. You get an increase in surface area, and that increase in surface area allows you basically to hold a larger amount of neurons. So I do believe that there are nice parallels in terms of surface areas. It's just not in terms of the whole brain. And that's where I think vertebrates and invertebrates brains varies a little bit. You have very discrete areas in invertebrate brains that are connected by axons, while in vertebrate brains, you do have also specialized regions, but it's just like one big mass of brain. So I think there are nice parallels. We just need to make sure that we're comparing analogous, or not homologous, but at least analogous structures in terms of their function, being able to understand patterns of morphological variation.
thank Denise again. Thank you so much.